So this presentation is co-authored by Kate Rinkovich, who's a graduate student of mine. This is not her thesis work. I've been a bad supervisor, and I've managed to distract her into a number of other things, and this is one of them. So I want to be very clear about what shelf life really means, because this was a fairly substantive issue under the softwood lumber dispute, and it's something that we have to keep in mind when we talk about things like shelf life. I define it as the post-mortality rate of decay and degradation of wood quality and quantity in these dead trees. It also affects the rate of change in stand structure. So not only are the trees themselves being affected as they change from dying to, or living to dying to dead, but also as they start falling out of the canopy, there are impacts on the vertical structure, which has a number of effects, things like wildlife habitat and fire susceptibility and other things. So having this knowledge about what happens after the trees die is important for strategic planning, not only around salvage harvesting, but also around land use plans and fire protection. So the objectives of our research was to really look at the relationship between time since death and measures of wood quality and quantity in trees that have been killed by pine beetle. And our goal is to produce some kind of regression model that enables that information to be put into things like what we just heard about. And we also wanted to try to quantify the influence of site factors on wood deterioration. How much does it vary from one place to another? And finally, to determine the rates of tree fall. The fiber itself, unless it's decayed by saprot fungi or eaten by wood borers, doesn't go anywhere. It changes dramatically in its characteristics, but it is there until the trees fall over and hit the ground, and then as the moisture regime in the tree increases, the saprot fungi and the wood borers take over, and that's the only way that the fiber is actually going to be lost. So the ultimate determinant of being able to use the fiber for some source, whether it's 2x4s or pellets or bioenergy, is how fast the trees are going to fall out of the canopy. So I want to mention the other shelf life factors, and these ones are extremely important. What I'm going to be talking about are not these things. I don't know anything about these things, but it's important that you keep this in mind in discussions about shelf life. So markets for the product. If you can sell the product, whatever it is you're producing at a high rate, then you can afford to spend more to produce that product. The cost of the raw material. In BC, we're having big troubles now with transportation costs. Operating costs. How much waste is there in the processing? What kind of equipment are you using? The manufacturing technology, and of course, log inventories. So that is not what I'm talking about, but I don't want to suggest that those are not important features. In fact, they're probably more important almost than what I'm going to be talking about. I'm limiting my talk to the biophysical changes that happen in the wood. This has been an ongoing project. It started quite some time ago, 2004, 2005, with a synthesis report where we tried to get all the existing literature out there on the so-called shelf life. Then we had some funding to do a first phase of sampling in British Columbia, and we thought we were going to access trees that had died early on in the outbreak, but when we really figured out how long ago they had died, we found out that that was only up to 5 years ago, so we went back to the Mountain Pine Beetle Initiative and got more funding, and then managed to find some trees that had been killed earlier in the stages of the outbreak development. So that's the second phase. And then the third phase is the work that we've been doing in Alberta. The first part of it, where we sampled in the boreal mixed wood forest, was funded by the TRIA project. And what I'm talking about, and I'm talking about that today as well, but the second phase of that in the Foothills area has been funded by the Foothills Research Institute. So here's the study areas for phase one. This is an area near Tweedsmuir Park in British Columbia, if you're familiar with that. So we sampled a dry subzone and a sort of a mesic or moist subzone in those samples. And in Alberta, as I mentioned, our first year of sampling here was focused in the mixed wood area, and then we moved into the foothills last year during our summer sampling. The variables that we look at are the response variables anyways are the moisture content both in the heartwood and in the sapwood, the wood density in the heartwood and the sapwood, and this would be an indication of how quickly saprot fungi are taking out the cellulose, the penetration depth of blue stain fungi, which is critical if you're producing dimension lumber for the Japanese market, the number and depth of checks, the penetration depth of sap rot, and the wood borer damage. And in our most recent samples in Alberta, we also included check length, and I'm sorry I don't have the results of that for you today, but we'll have it pretty soon. The predictor variables that we looked at were the date of mortality, or years since death, the size of the tree, different kinds of site factors, the soil moisture regime, the subzone that the trees are in, the height along the stem. So we sampled at multiple places along the stem. We wanted to test the assumption is the same thing happening at breast height level as up higher in the tree. And then also looking at factors to do with stand density. This was based on some information that we got from operators who were harvesting these trees who felt that in more open grown stands checking was worse. So we tested that assumption. So talking about time since death, again, it's another thing that we have to be careful about. A lot of people use external indicators as a way to estimate years since death, and that's what we did too, at least in our BC samples, where we tried to make sure that we had samples of trees that had died recently and then samples that had died quite some time ago. And so we used 4 categories of time since death, the 2 categories of red and then 2 categories of gray. And we also ended up with some green attack trees in there as well. So we first went in the case of all of the sampling that we've done, whether it's BC or Alberta, we have done plot level surveys or stand level surveys using plots to quantify things like the stand and tree attributes. And it also enabled us to select individual trees for destructive sampling. And we also took a snapshot in time of the fall rate in those stands. But then we get into the more detailed, intensive sampling at the tree level of those parameters that I mentioned earlier. And in BC, our initial sampling structure was based on ensuring we had an equal distribution of samples from 3 moisture regime classes, wet, mesic, and dry, 3 diameter classes, and 4 time since death categories, which I just showed you earlier. We kind of abandoned that approach in Alberta because I have to commend the Alberta government and industry for your alacrity and thoroughness in how you attack the earlier stages of the pine beetle infestation here. And we had a heck of a time finding trees that had died in the earlier parts of the Alberta outbreak. And so we were pretty much taking what we could get. The field measurements, we follow the BC Forest Service procedure for decay, waste, and breakage surveys, and that involves following the tree and then sampling at 12 different locations along the tree, 1 at stump height, 1 at the 10 centimeter top, 1 at DBH, and then 9 places in between. And in Alberta, based on what we had learned in BC, we actually only sampled at discs 1, 2, 4, and 8, which I have marked up there. At each cookie, we measured a number of things: the diameter, the blue stain depth, the depth of checks, the sap rot damage, and then we took pieces of the heartwood and the sapwood and measured their moisture content using the oven dry weight procedure. Now, to get the precise dates of when the trees died, we use a tree ring analysis. I'm not going to go into that in detail, but it enables us to take samples of the tree, measure the rings after going through a fairly intensive sampling or preparation process, comparing the ring width variation to a master chronology, and then coming up with an actual precise estimate or determination of when that tree laid down its last year of growth, which would be the year that it died in. So we have very, very accurate years since death data. If we could not accurately date a tree, it did not go into our data analysis. In the case of trying to determine how long it takes for these trees to start falling over, we had the opportunity to determine a snapshot at the stand level surveys. So every time we went into a stand to do a survey, we quantified the percent of the trees that were down versus those that were still standing. But that only gives you a snapshot. We also initially started looking at trying to find situations where a dead tree had fallen against a live tree, doesn't matter the species, and that live tree had a scar created on it. And using tree ring analysis, we're able to date of that scar. We started doing this, we had 30 pairs of trees from our first BC sampling, and since then, through funding from TRIA, we have expanded this project tremendously in BC, and we're looking a lot more intensively at different sites in British Columbia trying to quantify the factors that relate to tree fall. And again, I'm not going to be talking about that today because we're just starting with the data analysis. But we are following this forward, and the information will be available to you. So looking at our stand level results, you can see there's just a table here showing the time since death range, and you can see that it's quite limited for the samples in Alberta compared to what we've been able to learn in BC. So today I am going to give you some of the results from BC, and you can make a comparison. You'll see that many of the trends are the same anyways. And so this just gives you the percent down in the stand. So up until years, the stand has to have trees in it that were dead for quite some time before we started having any tree fall. And I'll talk about that a bit more later. In the Alberta stands, there's virtually nothing coming out of the canopy yet in our experience. One of the interesting things that we're able to do because of this ability to precisely date when the trees died is to look at the progression of mortality in individual stands. So in the drier subzone in BC, you can see that early on, so this is 1999, there's a few small trees that are getting hit. So this is before the epidemic phase has really started, and the beetles are confined to trees that don't have the ability to pitch them out very well. But then as the attack really gets motoring, it's the larger diameter trees that are hit first, and then progressing to smaller diameter stands or trees in the stand. Similarly, this is an example from the moist subzone. Now Alberta is showing the same pattern of smaller diameter trees being attacked before the beetle has hit an epidemic phase. There doesn't seem to be this decreasing diameter class selection, but it could be because we really only were able to sample a large number of trees that had died in either 2009, 2010. That's where most of our sample size was. So as the attack progresses, I would expect to see the same decline as the beetles are forced into smaller and smaller diameter trees. But we don't know that yet. The sapwood moisture content is probably the single most important variable when it comes to the changes in wood properties in these trees. Moisture content drives everything. It's the process of the trees drying out that creates the checks, but it's also a requirement for saprot fungi. If it gets too dry, saprot fungi can't do their thing, and they don't create decay. So I'll talk a little bit more about that later. So on the top here, we've got two graphs from the BC samples. And the way that you read this is disc 1. And so that's the leftmost bar in each of the clusters. And that's the disc taken at stump height. Disc 2 is the disc taken at breast height. Disc 4 is 1/3 of the way up the tree. And disc 8 is 2/3 of the way up the tree. So in the dry subzone, for example, we see a percent moisture content that is up around 80%. Actually, in a green tree, it's usually over about 100, even over 120%. And that reduces very quickly. Within the first year or two, the moisture content of the wood has dried down to roughly fiber saturation point. The exception is with the first two discs, and especially the first one, so the one that's closest to the base, where that tree is still able to passively absorb moisture from the ground. And that creates a completely different environment in that lower section of the tree than further up, and more on that later. In Alberta, we saw a very similar response, except this is in the mixed wood samples. I've got the foothills in the next slide. That even that first disc dropped down in moisture content very, very quickly. So just pay attention to the top row right now, because I'm just focusing on the sapwood. So again, the same order of discs. So this is your lower foothills, and this is the upper foothills. And this is a combined. So again, you've got a fairly rapid dry down to around fiber saturation point, with the exception of that basal disc remaining moist for quite some years post-mortality. In the case of the heartwood, which is already at a lower moisture content anyways, we see the same decrease, and again the same retention of moisture in that basal disc. In Alberta, this is again the mixed wood, a similar relationship. And now we focus on the bottom graphs here, and you can see that there's not a lot of change in the heartwood moisture content in the foothills area. It's already quite dry, and there is some effect of mortality, but not that much. When we look at the differences in moisture content of the wood that are due to different site factors, where we sampled in BC, as you saw on the map, we sampled fairly close together in terms of the dry and the mesic subzone, and we didn't find a lot of difference in the moisture content between those subzones, with the exception of, again, what's going on at the basal part of the tree. And it's only in the wettest ecosystems where there was a significant difference in moisture content compared to the drier ones. Most likely, if you were to sample in the central interior of BC and compare that to the area around Penticton, that we would see a difference here. But in the case of, within the confines of this study, we have not observed a major difference due to soil moisture regime. I put this in here. We don't focus a lot on blue stain depth because it's basically limited by the depth of the sapwood. We found that in faster growing trees, which have more sapwood, that's where blue stain tended to be more prominent. But I do want to point out, this decreasing trend that we observed in both samples in Alberta. It lines up with what we observed in BC, is that there appears to be essentially a fading effect of the blue stain with time. The reasons for that I'm not completely sure about, but it probably has to do with the reduction of volume of the hyphae of the fungus, which is what gives it the blue color. So getting into the checking question, which was probably the most important factor with regard to the softwood lumber dispute anyways, and I think it is the most important factor with regard to trying to manufacture something out of these trees, especially if you're trying to produce dimension lumber. Not so important for pellets and other things. So the percent of trees with one or more checks by disc height. So basically, what you need to take away from this is by year 2 to 3 or so, most of the trees have developed a check, and that is essentially what we found in Alberta as well. This is the mixed wood samples. So by the first year since death, between 30 and 40% of the discs that we measured in each of those trees had a check shown in them. If you look at the entire tree as a single sample unit, by the second year post-mortality, between 60 and 70% of the trees are checked, somewhere on their length. The number and depth of checks, these are from the BC samples. So number of checks up here, again, the same order of discs. So these are fairly small numbers, but keep in mind this is a check taken at a 4 centimeter disc. It's not representing the entire tree or entire log. And a couple things to note here is that there is an increasing trend with time on both the number and the depth of checks, and that the disc 4 tends to be the disc that shows the most checking over time. So it's the middle portion of the tree that has the most and the deepest checks. It's not the bottom portion of the tree because that area remains more moist and the sapwood has a higher moisture content, so it limits the development of checking. And it's not at the very top of the tree because it tends to be a smaller portion and it's not under the same torsion forces as the tree dries out. So what we found in BC and what we've also observed in Alberta, which I think is the next slide, is that the checking is worse at discs 2 and discs, that should be 1, 2, 4, and 8 up here. So that's disc 4, that's 1/3 of the way up the tree, and that's disc 8, 2/3 of the way up the tree. But again, we find that the worst checking is in that middle portion of the tree. There is an increasing trend with check depth and also somewhat with the number of checks. In BC, we found that the diameter at breast height was a significant indicator of the number and depth of checks, and we worked that into the regression models that we developed. In Alberta, we're not seeing that kind of a relationship yet, and I think it may relate to the thing I talked about before, is that we haven't sampled enough across a distribution of DBHs that are being hit by the pine beetle. Or it could be that diameter here doesn't matter as much as your drier atmosphere does, and it's that drying effect that is driving the development of checks rather than diameter having a big influence. We do see a little bit of a trend, and it is statistically significant with regard to diameter and check depth, but we're not seeing much with regard to diameter of the tree and the total number of checks that the tree will develop. We have more work to do on this analysis still. Looking at the percent of trees. So what we want to do is try to come up with, if a forester is standing in the field looking at a tree, normally they see around DBH, maybe a little higher and down to the base of the tree. What is it that they can expect to see over time? So this top graph here is the percent of trees that show no checking, but only at that 1.3 meter, 4 centimeter sample disc. So it doesn't represent what's going on in the whole tree. It's just what you would see at DBH. And we chose DBH rather than the base of the tree, which is represented down here, because of that profound difference in what's happening at the stump height compared to 1.3 meters because of the absorption of moisture from the ground. So this is a declining trend of the percent of trees with no checking evident at breast height. This is a sample size graph. You can see that we had some issues there. This, and the same graph is shown over here for the Alberta samples. This is only from the Foothills sampling. Percent trees with no sap rot in the basal disc, a strong decline. We're not seeing that in Alberta. We're not seeing as much sap rot development in Alberta. As you see, the numbers stay higher longer. And this is the percent trees with no wood borers in the basal disc. And again, there's a bit of a decline here. Most of the time, the wood borers that we're observing are carpenter ants. Sometimes ambrosia beetles, which we would find throughout the entire length of the tree height. Bark integrity has some effect. We looked at this mostly from the BC samples. So the bark integrity category here, these are trees that have tight bark, loose bark, and missing. And this is the condition of that, or the effect of that on the average number of checks and the average depth of checks. So as the bark falls off the tree, you can expect more and deeper checks to develop. Most likely that has to do with the loss of the insulation factor. As the bark falls off the tree surface, the sapwood is exposed more to the drying and wetting cycles, seasonal cycles, and especially development of small checks that happen over time, rather than those great big honkers that develop in the first year as the tree goes through a dramatic dry down. Seasonal changes in moisture content in the sapwood will also cause the development of checks, and probably the integrity of the bark, how tightly it is around the stem, will have some kind of does have an effect on the development of those surface checks. So looking at fall rates, this is again data from BC because we hardly found any trees that had fallen over in the Alberta samples. So this is a snapshot in time from the trees that had died between 6 and 10 years ago by diameter class. So not a strong relationship there, or not any. Using our paired trees, we get a bit more precise information. So this graph shows the years since death. So that's the black bars, and the gray bars are the years to fall. So most of the trees that we are included in this sample died between 7, 8, and 9 years ago. And tree fall doesn't really get going until year 6 post-mortality, but starts increasing around year 8. And I think that this is going to be highly variable across the landscape. And what we're trying to do now is to get a handle on what is driving that variation. It's not simple. It seems that diameter is important in some regions and not in others. That soil moisture regime doesn't appear to be playing the same role that we expected it to, but that's a project that we're still working on in terms of our data analysis. So the conclusions from this, the first one is that external indicators of time since death, red, green, and gray attack stages, are not very good at actually precisely identifying when a tree died. Just keep that in mind. It gets worse as you get into the later red and the early and later gray stages. It's very hard to tell when a tree actually died. Mortality in a stand can take place over a number of years, but the pattern of mortality may be different between BC and Alberta. We don't have the data yet to really address that. But in BC, we found that early attacks, the trees that are being attacked are the big ones, and then the beetles are forced into smaller diameter trees over time. Alberta, like I showed you, doesn't seem to be showing the same pattern, but it just could be that we were sampling in areas that just haven't experienced the full development of the local epidemic. Less than 1% of the beetle-killed trees fall within the first 5 years, but rates of fall start to increase substantially after that. at least in the area that we have sampled so far. The soil moisture regime don't really appear to have much influence on decay and degrade in BC, but we're seeing that Alberta, being slightly drier, does tend to have some differences in the extent and rate of development of checking. Most change in the dependent variables, so the development of checks, the sap rot, and those kinds of things, happen within the first 2 years. So there's a dramatic change as that sapwood moisture content changes dramatically, and then things tend to stabilize a little bit after that, but the number of checks and the depth of checks do continue to increase with time. Checks develop first and are worse in the middle parts of the tree, so that's pretty much from a little bit above DBH to about 2/3 of the height of the tree. Response variables depend with where you're sampling, and tree size seems to be a good predictor of checking in BC, but we're not seeing that kind of a relationship in Alberta, and again, it may be because the ambient moisture here is driving the system more than diameter. So we came up with a conceptual model following the sampling in BC, which pretty much, I think, relates here. So there's a window of opportunity that's very, very short, that goes up to about 6 months post-mortality, where you have green wood characteristics, and this is driven by the blue stain component. Between 0 and 2 years, there's a rapid change in moisture content in the heartwood, and that's when most of the checking develops. And there is some initiation of sap rot at this point. The sap rot continues to develop at the base because the tree remains wet down there, but everywhere else on the tree, the sap rot essentially decreases or stops. It just gets too dry for the sap rot fungi to metabolize. So there becomes a period of up to about 8 years of relative, and I emphasize that, stability in the deterioration of the wood properties. And then the trees start to fall over, and then the whole thing changes, because as soon as the trees hit the ground and they absorb moisture, then they become much more susceptible to sap rot, and that's when the fiber is actually lost. So I want to acknowledge the assistance that we got from industry, especially out of Grand Prairie, and the, I'm sorry I can't remember his name, but the forest health officer in Hinton was really helpful as well, as was the staff here at the Foothills Research Institute.